pharmaceutical drugs. So just to kind of give some background to the space, the delivery of nucleic acids is highly specific and selective, and many modalities, including plasmids, mRNA, siRNA, antisense oligonucleotides, and many of us have heard of CRISPR-Cas9s, are very important therapeutic modalities. They're important because they have the promise to repair the root cause of genetic disease. And by rewriting the genetic code, they can reprogram a patient's immune cells to potentially attack cancer. And we've seen this with some of the CAR T-cell therapies that have been very successful in the clinic. Also, one thing that is sort of an important area that I think is sort of up and coming is that we can have the potential to target selectively antibiotic resistance genes while leaving the healthy microbiome alone that could offer new and important defenses against untreatable infections. So currently right now in the clinic, and I'm going to talk a little bit about some of the FDA approvals, our most FDA approved therapeutics involve an engineered virus where researchers have taken the infective gene out and put in the therapeutic gene to actually deliver this into humans via various methods. Of course, viruses have developed the ability to deliver their payloads and their bad payloads into our cells over millions of years. So we've taken advantage of that as researchers, but there's problems that have come up with this. So just to give you some background on strategies and how nucleic acids are actually delivered, you can have what I mentioned, like the CAR T-cell therapy or stem cell transplant therapy is what's called an ex vivo therapy, where you can take cells from the human body, you remove them, so you can isolate immune cells or isolate stem cells. You can correct the genome in those cells and then deliver and do what's called an autologous transplant of corrected cells. So this is where you take the patient's own cells to do this. And there's less risk of rejection of these. So, like if you have a donor for a stem cell transplant, there's always the risk of potential rejection, despite them doing a lot of work to understand and make sure that the donor is a good match for the recipient. There's also what's targeted therapeutics, and that means, like, for example, systemic injection, where you can actually deliver and it has signals where it will target a specific organ. And there's a lot of work that have been successful at targeting liver for gene therapy. And I'm going to briefly talk about this. And then there's like sort of in vivo systemic, and we've seen this recently with the COVID vaccines, where you can just inject a formulation that contains nucleic acid directly into a tissue. Like, for example, the vaccine is directly injected into our arm, or you could administer directly locally to an organ. For example, delivery to the heart, you can inject heart muscle or inject various muscles, or even direct locally to, for example, the liver, spleen, kidney, brain, etc. As mentioned, there's many approved, and this just highlights a few of gene therapies that are approved. So this is a reality. Of course, the COVID vaccine has popularized this and was one of the first widely available sort of genetic-based therapeutics that was a vaccine, but was encapsulated by a synthetic carrier. Engineered viruses are extremely expensive and they take a very long time to manufacture. But as you can see, clinical trials in gene therapy, while they've basically focused on these viral vehicles, non-viral modalities are increasing in clinical trials right now. So that's something that's really important. You can see most clinical trials are focused on cancer, but there's many genetic diseases, such as genes that involve one or diseases that involve one gene, which are called monogenic diseases. And as mentioned, for example, infectious disease, as well as cardiovascular disease, which affects humans widely. Now, why are viruses problems? So what happens when you go through a clinical trial, you take out the infective gene, you put in a new gene, you have to go through the whole clinical trial process again for every time you do this for every therapeutic. So about 80% of the process is an FDA review time for gene therapies, and it's required to understand its manufacturing and quality control process when you manufacture these gene therapies. 30% of the product cost arises from this development. And so you can see how outrageous the prices of these gene therapies are. So Luxturna, which is marketed by Spark Therapeutics, has a price tag of almost $900,000 an injection, and Zolgensma, which is a pediatric gene therapy for treating spinal and muscular atrophy, it's a genetic disease, has a price tag of over $2 million. And I will say that my lab has a personal connection. One of my graduate students has a niece that actually has this disease and was administered Zolgensma early on. Shockingly, the insurance company did actually pay for 2 administrations of this gene therapy. So over $4 million for 2 administrations. Her disease is cured. However, if you imagine we have a lot of problems with insurance companies, this is unfeasible to make this widely available to the public. Also, viruses have a very long queue time, as long as almost 2 years just to get enough virus to run a clinical trial. Also, there's, in clinic, there's mutations that have happened. The virus causes immune response. So there's been a lot of problems where people have actually passed away in clinical trials. Now, the advent of non-viral, in addition to the COVID vaccine, has also been sort of bolstered by this very important New England Journal of Medicine article that was published in 2021 by the company Intellia. And what they did is they encapsulated a CRISPR-Cas9, which was an mRNA that coded for the enzyme, connected to a guide RNA that targeted a specific gene in liver to cure genetic disease, and it was encapsulated by a lipid nanoparticle carrier. So this is a synthetic lipid that was pegylated on the outside, and a lot of their work showed that the pegylation, so after injection, the pegylation actually binds to ApoE, and this opsonization of process causes ApoE to actually target delivery to liver. And so in this work showed the first CRISPR-Cas9 successful in vivo gene editing for a disease called transthyretin amyloidosis in human liver in clinical trials. And so this was a very exciting moment and that bolstered the interest and importance of non-viral, so synthetic carriers, for delivery of these very sensitive payloads that are important for improving quality of life of humans. Now, I've showed a few examples of sort of gene therapy, but with the popularity, the payload, which is the actual nucleic acid, that payload type is increasing in complexity. So we have DNA, we can have single-stranded DNA. So this is double-stranded. We have single-stranded oligonucleotides. We can have RNA instead of DNA. We can have antisense agents, which can be DNA or RNA, or even siRNA, or even various proteins that interact with nucleic acids. So for example, like delivering CRISPR-Cas9 protein instead of mRNA that codes for the protein. So the complexity of the payload is increasing because which means that there's a need for new carriers to, encapsulate these sensitive payloads. So the carrier, what it does is it basically wraps around the payload, and DNA and RNA and siRNA, if you just inject those into your body, they're very sensitive to nuclease degradation. So once they hit their target, or if they even make it to their target, often they're going to be degraded. So you need a carrier to protect this from enzymatic degradation, but also help circulate in the bloodstream target the specific cells and make sure that it's getting the payload into the specific cell and to the specific area in the targeted tissue. For example, many nucleic acids such as siRNA or messenger RNA have to get to the cytoplasm, or genes and CRISPRs have to get to the nucleus. So you can see that there's a multitude of extracellular and intracellular barriers that the payload packaging helps with. So from, just product storage and thermal stability to preventing various degradation routes, premature release, as well as preventing immune recognition. It also makes sure things get to the right target and make sure they traffic and are internalized and unpackaged in their payload. So this is a big job for a small nanoparticle that's essential for improving quality of these therapeutics in vivo, that again is making a reality of gene therapy, bringing the price way down so it is reachable to the general public and even developing nations. So my lab is very interested in developing polymers for delivery of nucleic acids. And, polymers have been the convention formulation vehicle for small molecules, protein drugs, as well as monoclonal antibodies. So the question is, why not deliver genes with these? So my group has been working in this field for over 20 years, and we've got a history of showing that we can package payloads, deliver payloads, target payloads, facilitate thermal stability of these payloads. And I'm going to show you a snapshot of this work that my group is doing, and then sort of tell you more about kind of where we feel like the area needs to go to make sure we can democratize this therapeutic strategy to the world. My lab, and just to give you an overview of my research lab, is a polymer design and development lab. And as Jim mentioned in the introduction, we have a lot of things we're interested in. Delivery of nucleic acids, understanding fundamental structure-activity relationships and applications is central to my program. But we also have programs in understanding delivery of small molecule drugs, for example, doing oral delivery, as well as just understanding methodology of polymer chemistry and making sure we're using green methods when we're doing this. And so using reagents that are non-toxic, environmentally friendly, and this translates to making sure our methods are friendly for, biomaterials development. So the overarching question that sort of drives my research lab is, can we create tailorable and affordable polymer vehicles for delivery of these very sensitive payloads? And in general, as I'm going to show you, we tend to make polycations, and these cationic polymers interact with these sensitive nucleic acid payloads just via electrostatics. So RNA, plasmids, CRISPRs, messenger RNA have a phosphodiester backbone that are negatively charged. And when you throw in a polycation to a polyanion, the binding is driven by entropy and release of counterions in solution, and we can spontaneously form these nanoparticles that we refer to as polyplexes. So again, polyplexes or lipoplexes with lipids have the job of protecting these payloads from enzymatic degradation, protecting product storage, for shipping purposes. And as we know, lipids are not very stable, so they are required to be stored at -80 freezers. And, if you're going to get your COVID vaccine, you got to make an appointment so they can thaw the formulation before injection. But this requires that the administration sites need this cold chain storage mechanism. And again, this is not accessible for many developing nations. So it's very important to develop these carriers that can facilitate stability, but also facilitate signaling for being transported in the blood and circulated in blood, as well as being recognized by cell surface receptors, internalized, and then ultimately delivered to their final destination within the cell. So my group, again, my students join my group and postdocs join my group because they want to learn how to make polymers. And we love making polymers, and we make all sorts of different architectures. So what this says to the public is that there's an infinite design space. So we can make homopolymers of just cations. Those tend to be toxic in vivo because the cation can disrupt cell membranes and, non-specifically bind to very important proteins and enzymes in the body. So it's important to sort of break up this charge density with hydrophilic units that are not charged. And I'm going to show you some examples. So we can make various architectures like statistical, where they're statistically incorporated, or perfectly alternating incorporated, or even blocky structures where you get a block of charge and a block of hydrophilic, which, when the block of charge interacts, you can have these core shell structures where the core is the cation binding to the DNA. And then you have a three-dimensional shell of a hydrophilic unit, or these sort of statistical units that create these condensed polyplexes where the charge is very close to the surface. But these tend to not be great for systemic circulation, maybe direct injection, where these would be better for systemic circulation purposes. And I'm going to show you examples of these. Prior to kind of going over some of the polymers we've made, I want to show you that and convince you that what's very interesting is that my lab has done a lot of mechanistic studies of how these polyplexes interact with cell surface membranes and how they're trafficked within the cell. And what we've shown is that, excuse me, these polyplexes actually go through similar endosomal pathways to viruses. So some get internalized via clathrin-coated pits, go to endosome and lysosomes, where some, we can bypass this endosomal pathway and actually bind to receptors that facilitate caveolae-mediated internalization. And they're transported to the Golgi, which is kind of like the FedEx or UPS in your cell, that then can signal various transport mechanisms, such as retrograde transport to the endoplasmic reticulum, which localizes it into the nucleus, which can facilitate nuclear delivery. So we've done a ton of studies kind of looking at which cell surfaces, like heparan sulfate, they bind to, what mechanisms they go through, and what vesicles they're delivered. And basically kind of telling you, this is one of many mechanism studies my lab has undertaken to kind of show that these materials actually signal transport via the same mechanisms as viral carriers. I get often asked, like, okay, once they're in the cell, how are they released? We've done a lot of fundamental work to also show that, and this was a particular polymer we made to facilitate understanding of release of the polymer from the DNA carrier once it's in the cell. And what we did is we made a polycation out of quinine, which is a natural product. It's actually the bittering agent in tonic water, and we directly polymerized this statistically with a hydrophilic unit. So again, a cation hydrophilic to sort of break up that charge density. This has a charge, but it is also can facilitate intercalation in DNA. It's fluorescent, but more importantly, it's Raman active, and this allowed us to directly probe via spectroscopy in real time in cell culture to show what the mechanism of polymer release from these cells. So sort of long story short, with these, we showed very effective delivery in a range of cell types, from skin cells like keratinocytes, various human embryonic kidney cells, and these are HeLa cells, which are cervical cancer cells. Just show that we get really effective delivery. We're delivering a gene that codes for green fluorescent protein, and you can see that we get almost all of the cells 100% transfected. And then what's cool is you can actually, via fluorescent microscopy, actually see this is a cell, and this is the nucleus. You can see the polyplexes directly in the nucleus because that quinine fluoresces. But then again, as mentioned, we have that ability, since quinine was really a beautifully Raman active species, to use Raman spectroscopy in real-time imaging and follow kinetics of release. And basically what we showed was that proteins can infiltrate the polyplexes and sort of peel off the polymers from the nucleic acid in situ, and shown that this sort of protein-mediated release was a way that we found intracellular packaging of these payloads with these polymer vehicles. So sort of, again, moving on to some of our more applied studies, what we've shown is that, if we make, instead of the statistical, we can make blocky structures. And so this is a study where we polymerized a block of a carbohydrate monomer. This is trailose. It's often used as an excipient in many pharmaceutical formulations and helps cryo and lyoprotection of sensitive payloads. But what we did is made a monomer out of it, and we polymerized it directly, and we can vary M and can control the molecular weight of this block. And then we co-polymerized it in a block with a species, this primary amine pendant, that at physiological pH is protonated and creates this cation block. So when you add this block polymer to a nucleic acid, for example, siRNA that's negatively charged, you can bind the siRNA in the center with the cationic block and then display the trailose on the surface. And through this, what we showed was that this facilitated really good cryoprotection. So we can lyophilize these nanoparticles or these polyplexes to dryness, and then actually rehydrate them, and we get nanoparticles back. So this is preventing lyophilization mediated aggregation, and we facilitate dispersion after rehydration. So again, this is important because this could facilitate a method to allow dry formulations of nucleic acids to be shipped. Right now, the convention is to ship frozen liquid samples, which, if they get thawed in that process, are actually, facilitate phase change and actually ineffectiveness of the vaccine. So what we then also showed was we bound this to a nucleic acid that codes for luciferase, and we showed delivery of this into liver. And this was a hydrodynamic injection, so kind of a fast injection that facilitates targeting to mouse liver, and we got really good gene expression. So in the mouse liver, this showed then that these particles were also non-aggregating with blood components, because if they did aggregate with blood components, they typically show up and get stuck in the lung. But we were able to facilitate liver delivery with these stable formulations that displayed this corona of trailose. So kind of dovetailing on that, showing chemical targeting of this sort of similar motif. So instead of trailose, we switched out the carbohydrate to an N-acetyl-galactosamine. So part of the rules of life that, researchers have discovered is that liver has a receptor on it called the asialoglycoprotein receptor that can recognize N-acetyl-galactosamine, or more commonly called GalNAc. So we polymerized a block of GalNAc, which is this targeting agent for chemically targeting liver. This block of charge, again, formed the complexes. We actually put on the end a fluorophore to our polymer. That's a near IR dye called size 7, where we could then, after injection, actually follow delivery over time of where these are going in situ and use through body imaging in the mice. So we didn't have to sack them at different time points to kind of follow where they were delivered. So you can see within 30 minutes after this is a slow infusion in the mice, we get delivery to liver. And then sort of after a couple hours, we start to get washout through the kidneys. And this just shows that a majority of the nucleic acid was delivered to the liver, not very much to the other organs, kind of showing that, not only can a carbohydrate facilitate thermals and sort of lyophilization stability, but also facilitate chemical targeting of delivery within in vivo in mice. So now, kind of in the last portion of my talk, I just wanted to mention kind of where we're going with this. So what I showed you was that, we have the ability to make a lot of systems, right? So all sorts of like block polymers, statistical polymers, and then we can make various architectures like micelles, which I'm not going to talk about, but I have a number of papers on this, as well as sort of polymer brush structures. So there's sort of this infinite design space of chemistry and architecture. And then there's all sorts of payloads that we can have, and one size is not going to fit all for these carriers. So where we're going is, we asked the question, is it possible to sort of start building models via machine learning where we might have the predictive capability to say, okay, for this payload and this tissue target, what vehicle is going to work best? We're taking baby steps to that. And I'm going to just kind of show you in this last component what we've done to do that. So my group has kind of taken a step back. And what we asked is, the question was, we can vary out cationic types. We can understand the chemistry of these, like the basicity and hydrophobicity. But then there's other hydrophobic partners. And so understanding hydrophobicity and incorporation and composition is very important. So even with like two different monomers, there's still an infinite amount of polymers that could be made, right? So there's like various distributions, whether it's block or statistical. There's different compositions of these you can make. So for example, low composition, like 25% of hydrophilic, 75% cation, all the way to like 50-50 or, to more of the hydrophilic species. And then there's molecular, there's length of this. So again, just kind of reiterating the large amount of parameter space to make these carriers. And like, how do you pick? And that's a question we're trying to answer with this. So we started this strategy several years ago, and Dr. Ramya Kumar was a postdoc in my lab, and she joined my group pre-COVID, and I gave her this project. And I wrote a DARPA grant, and they kindly funded sort of this idea and allowed us to purchase some instruments to do high throughput synthesis of these systems so we could build libraries of different polymer compositions, chemistries, molecular weights, and then sort of a workflow of binding these to nucleic acids, looking at particle sizes, analyzing pKa's of the polymers, understanding basicity, calculating log P, so hydrophobicity degrees, and then screening these in biological systems. So we developed like a really rapid assay, and in this work, I'm going to highlight some of the work we've done with CRISPR-Cas9 to sort of screen through which ones are good for gene editing. We can get structure-activity relationships and computationally analyze this data to identify hits, and then feed back into our model into the synthesis. And so this is, Ramya Kumar is now a professor, assistant professor at Colorado School of Mines and Chemical Engineering. So some of you might have met her or will come across her. So she's a really talented person that's building her own lab in this space as well. So just kind of showing our workflow is that, our initial combinatorial polymer synthesis and assembly is that we took 4 structures. So this sort of mini library or family of amines that varied in pKa and ranged from tertiary to primary amines. These were all commercially available. And we built our library with 3 different hydrophilic units. That's MPC, the zwitterionic species, is super hydrophilic, to this pegma, which is hydrophilic, to hema, which is somewhat hydrophilic. So just one hydroxy group hanging off. We have parallel synthesizers in our lab where we can make, do this. We do a controlled polymerization called RAFT. I'm not going to go through the mechanism here, but we can do, each one of these is like a tube, which is a batch reactor system. We have 2 of these, so we can make 24 polymers at a time. It's actually very easy for us to make polymers and analyze them. So we made a variety, or sort of a first library that contained all these amines and sort of all these hydrophilic units, and looked at various chemical properties and their effect on biology. So in this is sort of a chemical library schematic of what we made. Is you can see in these families, for example, we made families that where we kept the hydrophilic unit consistent. As you go across the row, you can see we've switched it out with different cations. And then what, like, for example, P1 through P4 means is we varied the composition within this. So from like 25% cation to 75% hydrophilic unit to 50-50 to 20, to 75%, 25, et cetera. And then if you go down the row, you can see we've kept the cation consistent, but we've switched out the hydrophilic unit to PEG and then here to HEMA. So again, decreasing hydrophilicity of the partner, varying pKa of the cation, so varying chemistry here. So we're able to take all those, and we're very lucky to be in a large department that has, NMRs, that have auto samplers. My lab has a size exclusion chromatography instrument that has an auto sampler. We have an automated titration unit, and then we can get particle sizes. We have a plate reader that does this, and then we do manual zeta potential analysis on these, but we can get all this very careful chemical characterization data for every single one of these units very fast. And this just shows we've made variants of all these from 100% cation to 0% hydrophobe, 75, 25 to 25%, 75%. So this initial data that we generated is for delivery of CRISPR-Cas9s, and this is an RMP delivery to make polyplexes. And we designed an assay that we were able to screen through. So we developed cells that had a M-cherry gene in their genome that was actually turned off. When you deliver a CRISPR-Cas9, you edit the genome. We designed it such that you put in an indel and it actually turns on the M-cherry gene. So when we get red fluorescent cells, that tells us those cells are gene edited. And so this was a way to sort of screen through a lot of our polymers. We also used some high throughput microscopy where we took pictures. We developed an algorithm to look at sort of differentiating cells automatically and calculating editing efficiency just through image analysis for these in like 96 well plates. So this is sort of all the collective data of gene editing for all of these polymer variants where we're varying the composition, as well as the type of polymer, as well as the amount of polymer in the formulation. And what you see here is that we're kind of getting one hit that's really good and several off hits, but just kind of focusing on this hit. So this allowed us to really screen through a lot of systems very fast and identify a carrier in the cell type that facilitates delivery of gene editing really well. So this one composition, this was one of those materials that had a 50% HEA and a 50% of one of those cations, delivered this super well. And you can see compared to many of the commercial reagents from lipofectamine 2000 to these CRISPR, lipofectamine, CRISPR Max, Jet, CRISPR Max, et cetera. And so through this assay, we were able to screen through, find this hit, and then actually verify this data through Sanger sequencing, and so genome sequencing. We've also developed this assay for delivery of various understanding like internalization. So we used a green fluorescently labeled CRISPR that we could see it in the cells. And we've looked at mechanisms and basically showed that the polymer hit, that really good polymer, delivers more DNA into the nucleus. And so that's what's something we were able to really look at and show. So what do we do with all this data? We get all this data, we aggregate the efficiency, and then we combine. So we take all of the chemical variants, as well as all of the parameters of biological efficacy, and we're doing some machine learning. So we first started with random forest classifiers, and now we're moved on to more of a SHAP machine learning that is derived from game theory to really tease out what chemistries and architectural parameters are important for delivery, toxicity, and uptake to understand and develop structure activity trends that can help us then predict formulations. So with this, what we've shown is that we can get, we can figure out and tease out what parameters are very important for delivery. And for efficacy for CRISPR-Cas9, we've shown that the Hill coefficient, so this is a cooperativity and protonation of that cation, is really important for gene editing efficiency. these parameters are not so important. You can see sort of the zeta potential is slightly important. Toxicity, what we showed was that how large, or the radius of hydration, the larger the particle, the more toxic they tended to be. And, pKa of the cation also had effect, slightly zeta potential and slightly formulation ratio. And then internalization, we get pKa being really important and zeta potential being really important. So, overarching theme is that, I've kind of give you, where we're going with this is that, we're exploring and exploiting parallel experimentation. We're evaluating sort of high throughput assays where we're trading off some resolution, but then we verify with a high resolution technique once we get our hits to extract design rules. And then now we're starting to learn from them. And that's kind of our next phase. And we're going to start publishing some work here in the near future with that. But with that, to sort of end a little bit on time, early, I want to thank my group and thank you all for your attention. And I'm very happy to answer questions.
Thank you very much, Professor Reineke, for a very, very interesting and timely talk. We have had some very good questions in the Q&A box, so I'll start with one that's in order as they came in. This attendee asked, RNA is considered to be less stable and prone to errors. Is this a concern in administering RNA-based interventions?
Yeah, that is a really good question. So I will say, as you can see in my funding, we work with companies that are very interested in RNA therapeutics. And what they have done, and some of their technologies, is that they chemically modify the RNA, either methylate or use thioester type bonds to facilitate a little more chemical stability of the RNA, such that it isn't as error prone once it is delivered. So that is something I think that these pharmaceutical companies have done a really good job, is chemical modification of the nucleic acid such that it's not affecting gene expression. Once, if you deliver an mRNA and it's expressing a protein, it's not affecting error in expression, but it gives us a little more leeway on stability that we can work with to further improve with our carriers.
Very good. Thank you. Next question is, can you discuss the pros and cons for systemic versus targeted delivery? Is there an immune response that would vary in the two cases?
Oh, that is a very good question. A very complicated
Maybe a little bit more than you want to tackle right now.
Yeah, and I think I could spend another hour talking about this. So there's positives and negatives to both, right? So like if you can formulate something that you can inject and it will circulate and target to the correct tissue, this is obviously easier for the doctor or the nurse, administering this therapeutic, right? However, what happens is it circulates in the body, the corona binds various proteins, and there is a huge, wonderful body of work understanding how that protein corona, can either help target and or you get this opsonization and it actually causes immune response. So there's some understood in that, there's some not, but I will say that, for example, a lot of pegylated lipid nanoparticles have been shown to bind that ApoE and target delivery to liver. So a lot of pharmaceutical companies think, okay, we have liver delivery solved, but there's nothing is solved yet for targeting any of the other organs. Now, on the other hand, you talk about, direct injection, like let's say somebody has a brain tumor, it's not easy to directly inject things into people's brains. It is done and it is often necessary. But, there's really frontiers of this area. For example, trying to deliver a nanoparticle through the blood-brain barrier, extremely hard, but of high interest to pharmaceutical companies as well as, NIH and whatnot. So I would say that is a really important, up and coming, very timely thing that a lot of people need to kind of start working on solving.
Okay, great. Another question is there a limit to the molecular length that can be stuffed inside the polymer vehicle, the polymer vesicles? And does that dictate the method, the mode of delivery?
That is a really good question. So what I actually didn't say, so I thank you first for asking this question. So viruses have a very discrete length of gene they can fit in their capsid. So often many genetic variants. So I work with some folks in our medical school, for example, that do a lot of work on collagen gene. Collagen gene is huge and actually cannot fit in a virus capsid. So the benefit of non-viral is that theoretically we have no limit of what we can package. So we can package small payloads to huge, molecular weights that are millions of molecular weight, base pairs for, to treat many of these diseases. The larger the payload, so the larger the unit, usually the lower the efficacy. So we've done some studies that we've published where, we have a cell type, we have a carrier, everything's consistent, but we've just gone from short DNA to medium to sort of long to huge. And as you go to longer payloads, cells take those up less. So understanding payload size in improving efficacy is, again, something that's very important to this area. That is a kind of an up and coming field of research.
Okay. Another related question about the molecular weight or the molecular size, this time not about the shell, but not about the fill, but about the shell. So the question is, what is the typical molecular weight range of your polymer used for the polyplex in applying for plasmids or for mRNA delivery?
Yeah, that is a great question. And what we've done, kind of some early work in my lab have shown that, molecular weight can affect toxicity. And of course, if it's huge, long polymer, it tends to have slower clearance. So it can cause more problems with toxicity. So typically the polymers we're making are about 30,000 Dalton, 40,000 Dalton. We tend to kind of stick in the 30 to 40,000 Dalton range. We do vary molecular weight to understand roles, trade-off on, higher molecular weight tends to perform better, but they tend to be more toxic. So you kind of got to hit that happy medium of good delivery and low toxicity. But 30 to 40 kilodalton is about where we're at. Ideally, and things we're working on right now is a lot of the scaffolds I showed you don't have an in vivo degradation mechanism, but would be like cleared through the kidney and urine, whereas we're starting to make more degradable backbone systems and understanding that.
Okay. Question about the commercial treatments that you mentioned. The question is, how successful is Zolgensma in treating acute muscular atrophy in children, if you know? Yeah.
So, again, I have one data point that's personal to my lab where my student's niece was administered this, and she is very healthy now. I think she was administered at about 2.5 years ago. So, long-term efficacy is something, of course, they're constantly following a lot of these patients. To the best of my knowledge, it's quite effective. But of course, as with every single drug, performance and efficacy and tolerability is patient to patient, is always followed, for these gene therapies, and it's followed very carefully. So the drug works well, which is why it's on the market, and Roche is actually, still manufacturing it. But some insurance companies are paying for it still, but it tends to be very good.
Mm-hmm. Okay, great. Comment here that our NNCI network, one of our focuses is foci is about measurements or metrology. So, what measurements do you think can help us understand what key parameters need to be measured in your delivery vehicles?
Right, that is a good point. So, where I feel the needs are in this field are for more rapid imaging. And so I know, like you and I, Jim, have talked about.
That's why you wanted me to buy a high throughput.
An operetta, yes. but like my lab, they're expensive instruments. So having these really expensive instruments in my research lab, it's a big investment. I have some, but like we can't afford all. And then, more access to in vivo imaging, I think, and just being able to pool formulations and actually instead of, injecting one formulation in a readout, one by one efficacy, being able to pool formulations, sort of pull out data from in vivo or, ex vivo work. So that's kind of a need. I didn't mention in my talk, but I have actually co-founded 2 startup companies in the space, one that's kind of focused on polymer formulations, one that's focused on lipid formulations and machine learning and kind of high throughput data analysis. These companies are both in Boston, but one is called Nanite and the other is Liberate Bio. And so we're trying to develop some of this, commercially. But, from a fundamental standpoint, having these tools and accessing these tools at the university is really going to be very important.
Speaking of tools, you mentioned, this is a question of mine. You referred to a couple of machine learning techniques that you're using. Can you comment just generally on the use of machine learning tools in this field?
Yeah, so my group is an experimental group. And, the caveat is that we're downloading like some of these tools off internet that, computational scientists have very kindly made available for free. Our next generation, what we're starting to work with is we have a huge and very strong computational sciences program here on campus. Our computational chemistry faculty here are very strong, and I'm starting to collaborate with the group and trying to, get some collaborations here. But, a limitation right now is algorithms that can extract data and trends from small data sets. So I'm showing you, we're doing machine learning on data sets that are like 200, 500 parameter space. Ultimately, these machine learning algorithms want like millions of data points, which experimentally are very difficult to access right now. So kind of the forefront of machine learning is a lot of my computational friends are working on algorithms for extracting trends and predictions from smaller data sets, and that's really an important and upcoming area in the space.
Okay, very good. One listener had a question about the intellectual property fight that's happening between Harvard and Berkeley. Are you familiar with the CRISPR IP controversy here?
Yeah, that is really, and I think they're starting to resolve it. I was following it very closely, and then it was just kind of like, oh my goodness, I can't I can't dig into it. But
it's for the IP lawyers, huh?
Yeah, it's it's unfortunate. I think they're going to resolve it, but it's a tricky fight because it's kind of like who designed it first, but they both published it at very similar times, and these things happen, like faculty are working on really cool stuff, and then all of a sudden, like, we've discovered the same thing, and first to publish or first to file the IP, that's really critical.
Right.
I guess long story short is always hedge on filing your IP, like file that invention disclosure and have your students fill it out. Yeah.
Absolutely. A couple more technical questions and we'll probably be at the end of our time. This person asks, for polymers that don't have a degradable backbone, do you have to keep the length of the polymer chains below a certain molecular weight so they don't stick around in the body? And if so, what is that value?
Yeah, good question. So as mentioned, we're kind of sticking to that 30 to 40 kilodalton. We have shown the smaller the molecular weight, the less toxic they are, but it just depends. But we have found that cells and animals can tolerate 30 to 40, but you have to sort of back off on the concentration of the formulation. So, you can have a long molecular weight and put less in the formulation, or you can have a short molecular weight and you can have a really high dose when you're administering it. So it's kind of important to like modulate both the dose and the molecular weight, but generally we're sticking to this 30 kilodalton, 40 kilodalton, but once we're making more of these degradable backbone, we might actually be able to hit higher molecular weights for administration with, it's something we're working on right now, so we don't know. Good question.
Good. So this last question gets at the fact that you're developing a lot of new cutting edge techniques here, and what's the time lag between doing the basic science, to validating it, to going to potential clinical trials, and all the way to the bedside? What's that timeline?
Exactly. So, this is exactly what inspired me to sort of form these two startups and form them in Boston, where it's the heart of the biotech industry and venture capitalist fund. I've been in this field since I was a postdoc, so it's almost, more than 20 years. And it's slow. And what has happened in those 20 years is that, gene therapy was hot, and then it was not, because there was failures in clinical trials. Then siRNA won the Nobel Prize, and it was hot and was going to solve everybody's, and then it was not, because again, things failed or were slower in clinical trials. Now, with the success of the COVID vaccines, the success of all those gene therapies, and there's many more it's here to stay. So pharmaceutical companies they have major investments. They're maintaining their investments in this area. It's not going to be fast, but I do feel that companies now have the tools to actually move forward with non-viral carriers, because every company I talk to wants to move away from viruses because of how difficult it is to manufacture and their immune response.
Right.
So we'll see. I know they've moved forward. There's a lot of success with lipid nanoparticle carriers. It's to be seen, TBD. We're just gonna have to watch to see when there are polymer carriers right now. There's a couple examples, but when that becomes sort of the next wave of success in the field. So it'll take a while. I hope faster. Again, the two companies are working hard on this that I founded, but they have very lofty goals. We'll see. Hopefully, we can make them work.
Right. Well, thank you very much, Professor Reineke, for a very interesting talk and very refreshing perspectives, and and enlightened and hopefully enlightened all of our listeners. To our listeners, I just wanted to let you know that we hope to have additional talks within this webinar series of addressing the rules of life. If you have topics that you would like to see addressed, or if you have speakers that you could suggest to us, please feel free to contact myself or Professor David at Northwestern, and we'd be happy to consider them. So to everyone, thank you very much for attending, and watch for updates on this area.
Thank you.
Bye.
Thank you so much, everybody. Bye-bye.